Ready to rock and roll? Were you recording? Yes. Yes. All righty, let's get going. There's a few rooms here. You know how to, are you switch screens?
That's all the navigation is, huh?
Well, Tom does that, I'll introduce Tom. So welcome everyone today to a special life sciences discipline seminar. Before I start, I'd like to acknowledge and pay my respects to the Burnet people of the Burnet Nation, who are the traditional custodians of the land where we are located, and pay my respects to their elders, past, present, and emerging. Well, I might have to be near the microphone.
Sorry, use the microphone.
That's all right. Okay. And I'd also like to extend my acknowledgement to individuals who are located online, so the lands they're located. So it's my absolute pleasure to introduce Professor Tom Hope, who's a frequent visitor to Australia. It's been a long time. The last time he was here was September 2019. And for those who don't know, Tom has very strong connections, I'll say that, to the Burnet Institute. We've had 2 PhD students from the Burnet that completed their postdoctoral studies with Tom. You might remember Jenny Anderson and Anna Figueredo, who was in my lab. I think Tom caught up with both of them yesterday. So Tom is a professor at the Department of Cell and Developmental Biology and the McCormick School of Engineering and Obstetrics Gynecology at Northwestern University in Chicago, Illinois. All the way from there. Chicago, Illinois, yeah. So Tom received his PhD from the University of California, Berkeley at the Department of Immunology, where he studied molecular immunology. He subsequently completed his postdoctoral training at the University of California, where he studied HIV molecular virology, and specifically the HIV protein REV. So some of you people who are retrovirologists will know what that is. So my first memory of Tom was many years ago at Cold Spring Harbor Retroviruses meeting, where he presented his pioneering work on engineering HIV particles with green fluorescent protein in order to visualize viral particles using deconvolution microscopy. And that was using, I guess, technology that was used by the Hubble telescope. So really innovative in its time. And this really heralded the concept of seeing is believing in the HIV field and beyond. And I, remember there were a lot of skeptics in the audience, Tom. There were the three guys up the front. They sit up the front as judge, jury, and executioner. Josh knows what I'm talking about, and very skeptical. What are those green dots? Are they actually viral particles? Well, I'd like to say, it's pleasing to say that Tom's work has stood the test of time, with many in the retrovirology field now using his imaging techniques today. So Tom combines molecular virology and cell biology approaches, as well as cutting-edge imaging to study HIV and how this virus enters the genital mucosa to establish infection. And he's advanced this work to whole body imaging and physiology, as well as omics. So he's got into the omics field as well. And this is to provide insights into HIV mucosal immunology, HIV transmission, and HIV cure research, which we're going to hear today. And Tom is a giant in the field, figuratively and literally. Tom's published in many respected journals in the field, including Science, PNAS, and Cell Host Microbes. And his impact in the field is immense. That's underscored by his publications that have attracted over 20, I looked up Google Scholar this morning, over 20,000 citations with an H index of 74. We're not allowed to talk about H indexes in Australia, but I'm going to anyway. The title of Tom's presentation is PET-CT Facilitated Multiscale Imaging, and Omics provides novel insights into HIV-SIV transmission, prevention, and HIV research. So with that, welcome to the podium, Tom. Over to you.
Thank you so much. It is such a pleasure to be back with my friends in Australia. I always treasure my opportunities to visit. I wonder if I can hear. we're not. And this first slide really shows kind of the scale or all the different imaging methods we are currently utilizing. And basically what's neat is you can connect them and do something called multi-scale imaging. And that I will illustrate later. But really there's up over here, there are, you know, the whole animal methods down to EM. So.
If you got it, slide there.
Yeah,
Just trying to make sure that what you're talking into the mind.
usually my voice carries greatly. So.
Possible to switch so that we don't have.
It might be that one. Well, anyway, you know, to be sort of involved in this HIV research, you can just click that one there. There you go. That's great. People don't need to see me anyway. It's scary. HIV science has just been, I think, fantastic, and I think we really showed our mettle for the pandemic, where the groups that made the vaccines and did a bunch of other things were the HIV researchers, and you know, I got to be involved a little bit, and we were excited because it was pretty clear that SARS coronavirus 2 was a punk, and we could beat it quickly, and you know, we made some mistakes. The biggest being that we knew about hesitancies and concerns and people sort of following good medical practices, and we should have gotten in front of that. But the science really was fantastic. So there's been a whole bunch of real progress in understanding this virus, and we're again a bit spoiled. So you're asking very basic virology questions about the coronavirus, like, well, how long is the replication cycle? We don't know. All these real simple things that we still don't know. There's a bunch of things yet to be done. The hard stuff is yet to understand. How does HIV cause AIDS? You know, why does disease persist with successful viral suppression by antiretrovirals, et cetera? And so my sense is that what we need to do is go beyond test tubes and tissue culture dishes. And without that context, it's going to be very hard to solve some of these problems. There's so many different disciplines and aspects of HIV. And the thing that's easy to forget is they're all happening at the same time. So somehow to bring all of those things together and appreciate them. So I really like Gilda's reference to labeling the virus and starting to be able to do that. We developed methods to actually watch how the virus interacted with mucosal barriers. And we kind of saw the virus was going everywhere. And so it became clear we had to figure out which of the paths the virus was taking into the tissue was going to lead to an infectious event. That meant we had to be able to do something that was kind of impossible, which was to find the place where the virus was. And I like to show this, especially for the younger researchers, because you sort of set your eye on a goal, and it can take a decade to get there. And this is a case of that. So we started with lentivectors expressing beta-galactosidase. We've all seen the tissues in mice, for instance, where the different tissues light up with blue, and you can get some information there. That failed completely. Then we tried fluorescent proteins, and that failed mostly, but we could find, after a lot of work, places where there were multiple cells expressing fluorescent proteins that were delivered by our SID-based vector. And so that, when we found one, we found several, so that gave us encouragement to put a beacon in there, which was luciferase. So if you read the luciferase literature, they say you can detect 3 cells maybe in a mouse. Definitely can detect 20. And so that started to work. And so we were able to use these replication defective vectors. The downside being it could really only show you the first cells that were getting infected. That was informative, but there's a whole lot left behind. We thought about this and realized that what this vector was doing was sort of mapping the places of mucosal barrier where a virus could get through. It was sort of mapping barrier dysfunction. So we mixed together real virus with the vector, and sure enough, as published in Cell Host Microbe, that told us where the real virus was. And so we were able to do quite a few things with that approach, but there's limits with light. You know, try to look through your hand. You can't see the light come through. Tissue wins. when it's having a fight with light. But then starting in 2017, we switched to PET-CT. PET-CT is a radioactive based approach, and it goes through tissue like it's not there. So suddenly this limitation was done, and this was really, I think, has become, I refer to it as a superpower. You sort of, like Superman, you can look inside of the tissue, you can see what's going on. And we're getting, my lab and team are getting really good at sort of leveraging this to ask different sorts of questions. So, what am I doing here? Sorry. So the idea is that, to understand what's going on in these systems, we need to have considerations of anatomy and physiology. And so that is going to allow us to gain some interesting insights. And that is going to be sort of the theme of this talk today. So, so we knew these were going to be rare events. And so how do you find rare events? You just need to know where to look. And so it was this idea of beacon guided necropsy beacon guided dissection, first with this this vector would make luciferase. Then we could go in and find the specific transduced cells with cherry, and you basically take because of the issues of light getting through tissue, we would have to take the tissue out, soak it in luciferin, inject it with luciferin, and you could start to see these signals. This is actually a site of a biopsy where the virus could get through. This was sort of important control early on. You then cut these tissues up into smaller and smaller pieces. Here you have like about 0.5 centimeter squares of tissue, and then finally down to 2 by 2 millimeters, and where the photons were is where we found the action. Oh. Uh-oh. Well, all right, so so using this approach, we were able to identify the initial cells that were getting infected. This could go one round of replication. And what we found was that Th17s are actually the favorite initial target of the virus. And then the second favorite target is immature dendritic cells. There were some undefined cells relative to their populations within the tissues we were looking at. And it sort of made sense. The immature dendritic cells and Th17s are metabolically active. They're in a daily way, they're surveilling the local environment, just local antigens, and whether it be commensals like the microbiome or even host peptides. And they just do this every day. And they're sort of like, okay, today's the same as yesterday. Don't worry. But if anything changes, it's sort of like this signal that something might be happening. They sort of put everything on alert. And very often, and there's going to be a lot of this today, we see things that aren't supposed to happen, that aren't consistent with what everyone believes. But luckily, this was one that everybody was ready for. So the Th17s were known to be depleted during acute infection in the blood in both man and cats. We knew these immature dendritic cells were infectable by the virus localized in mucosal tissues. And again, I think the key is they're metabolically active. Most of the T cells, for instance, are in a resting state. So, basically, getting a bit frustrated with the light issues and the ability, the limitations of that, we started to look at PET scanning, and this just illustrates the same sort of approach. We would deliver, we still depend on fluorescence. So for instance, in this first experiment, we fluorescently tagged antibody and then radioactively labeled it and instilled it rectally to see how it was being distributed. You can see then that signal within the colon and the whole animal. Then we can take it out and see this focal expression patterns. We dissect this tissue, put it into cryomolds, and then you put the cryomolds right through the same scanner. You get a result like this, where you have, there are 2 tissue blocks in every one of these squares. No signal here or here. If you're randomly selecting places to look, you could be looking in the wrong spot. However, you pick a hot one and then you go in and look for your fluorescence, and we can find it pretty easily. So, so this presentation here is going to illustrate sort of this idea of the why once you know what the piece of tissue is, you can start to do some interesting things. And so here we have an animal that has the distributed signal in the gut. This is actually an antibody injection. We can go into, say, the rectal, the colon, and see the site of bioluminescence. You can dissect out that piece of tissue and process it, for instance, for an injected fluorescently tagged antibody. and see how the antibody is distributed within the mucosa at different time points. You can take the same piece of tissue and process it differently for immunofluorescence staining. And so this is 4 days after a rectal challenge, and we can stain for SIV and CD4, and we can find infected cells within these tissues. You process that piece of tissue in a different way and do EM tomography. You can start to see this, which is an infected T cell 4 days after challenge, producing viral particles. So the EM gets you down to the nanometer range. The PET-CT is sort of starting in the meter range, and so that's 9 orders of magnitude of information going from the whole animal down to the viral particle in a single experiment. So this really, I think, provides us with a nice platform to begin to understand some of these processes. So about 20 years ago, I went to a NIH workshop and they were talking about PET-CT, and I just was like, oh, this is it. I got to do this. And then you need a cyclotron and you need a scanner, and you need all these things. And it just seemed impossible. But over time, different primate centers and things have acquired this equipment and the appropriate expertise. And then I found these fantastic collaborators, especially Francois Billinger and Mary-Luz Arianga, a couple of veterinarians, and they work at the New Iberia Research Center, NERC, in rural Louisiana. And so basically labeling different molecules of interest with copper, which has a 12-hour half-life. You can follow it for 3 days maybe, or with zirconium, which has a 3.3-day half-life. So we can follow it out easily to 2 weeks and potentially to 3 weeks. We can begin to start to ask some really sort of interesting questions. And really, I mentioned this earlier, but it's just the eye-opening superpower of PET-CT has really transformed the science of my lab. And the neat thing is you do this study and you scan the animal from the tip of their head to their toes. And so it's just this unbiased screen of where is the signal. And if the signal's in the big toe, it's nobody would have thought to look there in the first place, but you have to chase the signal. It's not influenced by tissue. You can do repeated scans, so you can get sense of dynamics and kinetics. Different probes can ask different questions, and we can really start to visualize the impact of anatomy and physiology on these biomolecular dynamics. And so at this point, we have 3 different labeling approaches. One is to label the viral particles. This is for the most part published, so I'll go through this quickly. And again, the key is there's fluorescent labeling here, this photoactivatable GFP. That helps us to confirm that the signal we're seeing is an intact virus and not some debris left from the virus that was eaten by a macrophage or something. The other area that has become very interesting is just taking antibodies and labeling them and watching how they distribute. And antibodies, monoclonal antibodies, are the primary sort of the platform for a lot of recent drugs. All of these sort of treatments for things like, from asthma to psoriasis to all kinds of things are treated with these monoclonal antibodies. And then, and what we've learned is that this FC portion really dictates where the antibody goes. And then subsequently there's function that happens later. So to study antigens bound by the antibody, you really want to use an FAB2. You keep the bivalent binding of the antibody, but you've cut off this confusing FC portion that is sort of competing with the antigen, and it always wins. There's always a lot more places in the host for the antibody to bind to the FC than there are antigens present. And there's a lot of logistics here that are considered. The CT scan takes 90 seconds. The PET scan itself is 15 to 20 minutes. And then you have other challenges like hot monkey excrement and things that just make it a big challenge, especially in these days. When I was a graduate student, we used radioactivity every day. Now it really isn't done very much anymore. We don't have a license to work with radioactivity, but New Iberia does, and you just have to wait 10 half-lives, and then it's not radioactive anymore, and then they can ship it. So this is an example of some of the work that we had done with labeled virions and found some interesting results. And so here, 2 hours after instilling first IgA and then viral particles, we can see that the particles actually are greatly distributed through the colon up into the transverse colon, and this was really very unexpected. We can then, again, take out these pieces of tissue. We can look at antibodies that we introduced and see how those antibodies are interacting with the virus that we've introduced and the antibody that we introduced. So in this case, what we did was we freshly tagged IgA and found it was doing some unexpected things. The IgA is blue here, the viral particles are red, and this is an example of immune exclusion. The virus is getting caught, trapped in the mucus above the barrier, and it can't get into the tissue. And this is sort of neutralization by sequestration. So the other surprise was we found 2 hours post-introduction viral particles in draining mesenteric lymph nodes. That's really very fast. And with the photoactivatable viruses, we could show that they were there and intact viruses because they were able to photoactivate. And really, this was also some very interesting insights. So this is just a little bit of that data, and what we could see was, and again, unexpectedly, and we're almost ready to start talking about this, but that when the antibody was present, the virus actually went more into the tissue, and that's because it's sort of, in this case, I looked at the textbook, and in mice, it's known that there are these CD123 dendritic cells that grab things and then take them to the lymph nodes. So we could see evidence of that, and we could also see that, again, the presence of the antibody actually allowed the virus to go further up into the gut. And then also it was when you had antibodies present, the more virus could get into these lymph nodes. So this was really very interesting and insightful. We did some other studies trying to gain insights into acquisition through breast milk in infants. This is a really important problem. Sorry, it's a little early morning for these pictures. And so what we were able to see was that very quickly, within 4 hours, the virus kind of made it completely through the animal and was coming out the back door. And that was a bit of a surprise. And then when we were looking at where infection was taking place, we found that it was in the gut. And so we know the gut is highly susceptible to infection because of the mucosal cells and things that are there. And so it was a bit of a surprise that that we were seeing infection. So we feel that this data is suggesting that, unlike what you would see in the literature, that it's tonsils and things in the mouth that are leading to this. It's actually the virus kind of getting all the way through into the gut where the abundant cells are and then infecting. And there's some other differences in younger humans and animals where the cells are all active and dividing, just trying to fill the body up with lymph. So now we'll move on to following antibodies. And when I started to do this, we started to do this, these studies. And I think many people still think about this way. It's sort of you inject some antibody IV, and it's a little bit like dropping ink into a beaker. So it just diffuses away, and over time it kind of spreads throughout. That's not at all how it works, actually. So this is This is an image I'm going to show you from the SuperPet at UC Davis. They have an array of scanners, and they can take that image every 2 seconds. And so you're going to see this is down here. You can see the signal, which is the femoral artery, and they're going to introduce fluorodeoxyglucose. And you can see that it basically quickly enters and goes up the femoral artery, hits the lungs and heart, and boom, it sort of blossoms. If you're watching the time, that was 35 seconds. And now at 1 minute, it's kind of distributed throughout all the major pipes in the body. From there, the antibody now starts to use different mechanisms to go into different tissues. This is at 24 hours with a fluorescent antibody, mucosal tissue. And you can see that there is antibody present, fluorescent antibody present within the arterioles and venules. And then even the capillaries are kind of really small, so it's hard to pick up that signal at 24 hours. By 72 hours, this is going to be a light sheet imaging and a more Z series and a reconstruction. But you can see that by 3 days, the antibody is coming out of the pipes and getting into the tissue. And we're finding that each tissue has different methods or different mechanisms behind distribution. And we're finding some really sort of crazy things, unexpected things that I'll be sharing with you today. But one of the first lessons was that it really takes about a week for the antibody to completely distribute throughout the system. And this is very contrary to a lot of the studies that were done for antibody development, vaccine development, because you would have these very expensive antibodies, inject them into the animal. You can just see it going out of the blood. It's like, hurry up, the antibody's disappearing. It's not disappearing. It's going into the tissue where you want it to be. And so I sort of wonder if some of those early experiments really missed something important by not letting the antibody get in front. And in fact, it's known that today that you can infect the animal and inject the antibodies a day later and still stop the infection in some cases. So we were moving along here and gaining some interesting insights, and the antibody thing was a bit outside of our main focus, but then the pandemic came along and we were approached by NIH to start to get involved with Operation Warp Speed to try to get a sense of if convalescent plasmid was a real thing or not. And so we wanted to see how those antibodies distributed. And up until this point, we had worked with 2 antibodies that mostly we used because they were generously provided by the Vaccine Research Center at NIH, BRCO1 and a modified version I'll talk about in a second. And we saw differences in those 2. So we like, okay, these antibody distribution has 2 phenotypes. Now we looked at 4 antibodies, convalescent IgG pooled from 30 infected individuals, normal IgG purchased at Sigma from a single donor, and 2 monoclonals to the spike. And suddenly we had 6 phenotypes. So that kind of opened our eyes that there was a lot more going on here. And so we can, as I showed earlier, we can do the PET scan and do it repeatedly over time. We can do light sheet and see how the antibodies distribute at tissue level. We can do fluorescence microscopy here of the lung and see where the antibodies ending up and which cells are interacting with it. But we also started to do analysis of various fluids to get a sense of, for instance, a bronchiolar lavage, which was one of the ways that you quantified the coronavirus. And so we began to do that, and that turned out to be, I think, a really important tool for us moving forward. So with just fluorescence, we sort of, and this idea takes a week. One of our first observations was that the antibody in wet epithelium in the female reproductive tract, the vaginal valve and ectocervix, is actually coming from the bottom up as the tissue differentiates and moves up. You can see from 24 to 48, 72 in 1 week. It takes 1 week to get distributed complete throughout that tissue. So there's sort of this tissue loading phase. This is known in the literature. And then once you achieve this steady state, some of the antibody is going back into circulation and sort of going around. And there's a lot of these cycles. And so if we looked at the fluorescent antibodies that are present here, for instance, in the mucosa, what we can see is that the antibody is concentrating in these basal epithelial cells, some of them, and then they differentiate up and basically die and form the dead layers of our skin. And so we can see this happening here in these sorts of images, and this quickly. So we wanted to be sure that this was not a consequence of our methods of labeling the antibody or something. So we could take the same tissues and stain for endogenous antibodies to more or less get the same results. We can see this subset of epithelial cells that have taken this up. I took it out for time, but there was a paper published in 1979 where they stained these tissues with EM methods, and they were like, there's serum proteins in some of these epithelial cells. What is going on here? And so again, I think that's just an example of sort of unexpected mechanisms of how these antibodies get distributed. So because of all of the value of therapeutic antibodies, they're being modified in a bunch of ways. And one of the most important ones is this mutation called LS that causes the antibodies to have a better PK, to persist in the blood longer, although that's sort of a perilous goal because antibody in the blood isn't what you want. You want antibody in the tissue. And so this is mostly expressed in capillary cells. There's macropinocytosis that takes fluids out of the blood, goes in, and when it acidifies at the acid pH, an antibody IgG or serum albumins will bind to this and be salvaged out, either released back into the blood or on the other side of the cell into the tissue, and then everything else that's left behind is turned into peptides by the lysosome. And this system is really quite amazing. Because it can, one of the ways to, for instance, treat autoimmunity that's antibody-based is you take bulk human IgG, inject a bunch of it into somebody, get their antibody levels up 5 times normal, in days, and definitely by a week, that all goes back to normal. And you've degraded 80% of your antibodies, the good ones, the bad ones. Your body keeps making more anyway, so you can reduce the pathology of the autoimmune disease. And you can see very nicely in these sorts of studies that the change to the LS makes these antibodies stay around a lot longer. And so you have to use less antibody. So what we could do is fluorescently tag two different antibodies, inject them into the same animal, and then look at them simultaneously, as you can see here. And the LS sort of had this very different distribution than the regular version. And so this was the antibody accumulating in the capillaries and circulation that were there. And so it really was neat to be able to sort of look at this textbook here, a schematic, which is showing the macropinocytosis. You take the two antibodies, red and green, in together. They then get sorted. The red gets kicked back out. The green gets degraded, and that is happening in the in the recycling endosomes. And you actually see these antibodies being separated in the brain of a capillary in the brain of a macaque 24 hours after injection. So we really, unfortunately, when we started this, there were not very good reagents that, especially antibodies that could bind to FcRn. But we were able to, basically there was a therapeutic antibody that bound specifically to the part of FcRn where IgG binds, so it still would bind BSA or serum albumins, but not IgGs. These things get patented, so the sequences get are out there, and then there are companies that will make these for you for a ridiculous amount of money. And so there's sort of this underground antibody work, but that was led to success. We actually have two antibodies like this. And so we could start to show that we were in fact in the textbooks again. So here is FcRn staining that overlaps with this VRCO1-LS, and that overlaps with this VWF protein, which is a good marker of capillary. So again, in the brain, you can see overlapping of all of this. So, again, I guess a lot of our interest is trying to understand these mutations that are put into antibodies that make them work better therapeutically, and then trying to understand the underlying mechanisms. So now back to the PET scans. And so we can do sort of, there's a zero time, which essentially means you inject the antibody and then do a scan. So early on, then at 2 and 4 hours, this is really showing how the antibody is distributing in the vasculature in the blood. And then as we get to 1 day to 2 weeks, we start to see this distribution. And this is an illustration of how different it can be. You can see that this pooled convalescent from 30 people, IgG, is going right to the liver, accumulating there and staying there. And then one of these monoclonal antibodies really never gets in the liver at all. The other thing is that, like so many methods, it's kind of your scaling and things is important. So you can change the scaling, and when you have a bigger range of scales, you have weaker, don't see the weaker signals, but you can gain some insight. So, and this very much was similar to what we had shown or could see for the distribution patterns for the fluorescent antibodies. So then we began to do these sorts of studies where over time we would do multiple PET scans early and late. We can then take these PET scans and the software. This is all sort of clinical software, and these are all tools that are used in the clinic every day. We can use the skeleton or use other CT aspects to draw circles around the area to segment the areas of interest, like the lungs. And then we can look at the signal that's within that, gain some interesting insights. And then there's also always caveats. So for instance, the zirconium can come loose from the chelator that's on the antibody, and it accumulates in certain bone plates. So you can see these signals in the wrists and in the shoulders, for instance. That's not where the antibody is going. You see that same localization if you just inject zirconium by itself. So we could then isolate these various aspects of the respiratory tract. Again, we were focused on COVID. We can plot that and see these differences between the different antibodies. We then, can take these signals that are in the different fluids, and this just shows a bunch of different animals and antibodies, and then different secretions and excretions from feces and urine to saliva, blood, etc. And then if you divide the signal in the PET scan by the signal in the blood, and you can even get this from the PET signal that's like in the heart valves where you have a lot of blood. You can get this sense of extravasation in tissue. How fast does it move in? And so when we look at it this way, it's actually the normal IgG that's moving most rapidly into the tissue. And this is an important way to look at it because the lung is this very, there's very little tissue there. There's a lot of vasculature. And in fact, that vasculature sort of dominates in terms of the signals that are there. So by dividing the PET signal by what's in the blood, we can get some interesting insights into how they're moving into the tissue. So we've now looked at quite a few antibodies. This list even needs to get bigger. We've looked at all IgGs for the most part, humans and macaque antibodies, polyclonals, monoclonals, and then different modifications. And each antibody shows a reproducibly variable distribution in PK. These differences are very reproducible between animals and different lots, and are really providing some interesting insights. So this is just a little taste of it. And you know, these are the initial observations, and then we have to drill down and try to understand them. So some of the antibodies go right to the spleen and stay there. This HGN 194, a not very good neutralizing antibody to HIV. So at 7 days and at 2 weeks, you can see the signal is really accumulating in the spleen. Other antibodies don't do this. For instance, BRCO7, 523LS, we'll be talking about this one here for a second. There really isn't this accumulation in the spleen. But the part that sort of vexed us was you can see the signal in the gut. And there were these gut-associated signals. They were very reproducible, very confusing. And at times, there was just a ton of signal in the guts of these animals. And we started to also look at some details. And one of the things we saw was this strange, unexpected signal here, a focal within the liver. And very often this was associated with LES or the LF mutation. We can rotate some of these here. You can see this. It's like I just have spent so much time learning about anatomy the last few years. And anyone want to guess what we're looking at there? The gallbladder, absolutely. You're going to get everybody excited about bile and gallbladders. So get ready. And nobody ever says that. So. All right. There you go. Did you keep it? Oh. Anyway, I'm sorry. So what we can see here in 4 animals that received this antibody, that the signal goes right to the gallbladder. It stays there, that bright spot, and then it ends up in the gut. In this case, in the transverse colon, but often in the ascending colon, and it concentrates there, and then it disappears from there, and then sort of you know, the signal changes, and it's like, well, what happened? So I asked Francois and Mary-Louise, I'm sorry to ask you this, but can you collect the monkey poo? Because we have to see if the radioactivity is ending up there. So they bring out this box of monkey poo. And I'm like, I'm sorry, I didn't explain this right. We need to every day collect a little monkey poo and see what the radioactive signal is. And there was no radioactive signal in the poo. So it's like, okay, what's going on here? And we saw this and kept looking at it. And it was sort of driving me crazy. I love these sort of mysteries. And so Francois and I decided to do, I think, a really definitive experiment, which is to inject the antibody, not in the IV and see some of them go there or not in the vein, but to inject it directly into the gallbladder. So with ultrasound, you can see the gallbladder, and we could do that. And just a little bit of review. So bile is produced in the liver and stored in the gallbladder. Bile plays an important role in digestion of fats and everything. These bile salts and things that are there are being salvaged in the gut as it moves along and then sent back to the liver. Biology is efficient that way. It gets introduced into the duodenum kind of after eating. And then it's known in the literature that dimeric IgA is actually captured by the IgA receptor, the secretory component, out of the blood and sent into the system and dumped into the gut, where we usually think IgA should be. And that's why there's very little dimeric IgA in the blood. So here is an example. And so we can see here at 30 minutes, because they inject it in basically the surgery room and then run them over to the PET scan. And we can see that very quickly it is coming out of there. It's starting to be distributed. We see this very interesting localization or concentration in the ascending colon, like we did for the IV injection. And then in talking with the vets and the people in the lab, they're like, those focal spots are lymph nodes. And it's like, what? And we got these results. And then I went home and just had this amazing weekend of trying to figure out what the heck was going on. And so very quickly, we were able to see that there's a subset of these lymph nodes that basically drain the gut viscera and then come up into the system in lymph. And one thing I didn't mention here, I'll go back, is this very sort of strange signal up here above the heart, right? And so that is actually, one of those signals is greater. It's the lymphatic duct, which is on the left side. And then there's actually, I didn't even know about this, there's sort of a right lymphatic duct that drains essentially this green part of the body, this smaller area, so it would pick up less of that signal. And so as we began to look at all this in detail and sort of look at the signals of, it didn't show up in the feces until 48 hours, some of the antibody, but it was showing up in the blood very quickly. And so what's going on here is that this antibody injected into the bile is kind of going into the lymphatics and accumulating there. And so maybe this is a way to get antibodies into the lymphatics and keep them there. And this could be very important for a variety of things, including cancer, because we know that metastasis is taking place through the lymphatics. So you can get your antibodies where the action is, you could maybe have an impact. So we've been doing a bunch of these sorts of studies, and I will say that, and I'm not doing this myself, but injecting things into the gallbladder is not trivial. So the second experiment, we call it OOPS experiment. They hit the liver, but missed the gallbladder. So you actually have this hole here this time. So that's the gallbladder without the signal, but we could see that it was distributed. And we actually, it's very rare that you can have the OOPS experiments, and we have a number of them that I'm not ready to talk about yet, but what we've learned is we can inject these antibodies in different parts of the body and they do very different things. And I think this is going to get really, really fascinating and interesting. So I think this is just a really nice example. And again, we can quantify this. This now has a very different behavior, right? It ends up in the blood immediately and has sort of different dynamics. And we've been doing quite a few of these, trying to get a sense of what's going on. This was the second experiment that went well. Hit the gallbladder, was in the bile. It's getting into blood with similar dynamics, and we see these very different sort of results that are coming out. So this is ongoing. The next place we're going is the brain, and it's going to get really very interesting. So we can follow these different antibodies and their PK by the radioactivity, and what's interesting is we can then get this sense of their PK. IC50s, and what's neat is the longer you look at these PET scans, you start to see more and more things. So there's a correlation with the signal in the heart with the amount of antibody that's in the blood. So PGT121 is leaving the blood pretty quickly. It's disappearing from the heart between 4 hours and 24 hours. This one surrounded by yellow is VRC07. It's kind of last seen at 48 hours, and then the one that has even better PK, we can follow it out in the heart into 7 days. So there's all kinds of, the more you stare at these PET scans, the more things you can learn. And I've spent way too much time staring at these, but I'm starting to really understand some of the dynamics here. So these antibody distribution is reproducible, and it's impacted by physiology, leading to a little bit of variation between animals, but overall very reproducible. Different antibodies are going to different places. I don't have time to go into this data, but a lot of it is being dictated by the glycoform. There's a glycosylation on the FC. Everyone knows that glycosylation is impacting interaction with different FC receptors, FC gamma receptors, but it also seems to be functioning as a zip code that says go to this tissue predominantly, et cetera. And these LS antibodies tend to go into the bile, and understanding all of these interactions is going to help us to better optimize therapeutic antibodies for a whole bunch of purposes. So we're going to now shift to the work to try to understand HIV reservoirs, SIV reservoirs, and cure research. This is the one that I really get in trouble with, but anyway, because nothing matches with what everyone believes. So, but we sort of did our transmission studies, and in many ways, the rebound, when you stop ART in somebody, the virus comes back very quickly, and it comes back with similar kinetics to transmission, for instance, and almost everybody comes back with this very quick return. It's a very rare individual that takes years or months or years before the virus comes back. So we wanted to, understand, think, where is this virus coming from? How is it maintained? What are the characteristics of the cell, et cetera? And so we were set up a system first with what's known as the early reservoir, where you challenge the animal and then start ART at day 4. And the reason we did that, it was sort of unlike what happens in people, although these days now they start ART as soon as you know you're infected. So it's better reflecting maybe the current times, but you don't get any immune responses, no antibodies, no cytotoxic T cells. So that was going to take out some variables. And then the biggest variable we were worried about is it's known that these defective genomes and things accumulate over time. We didn't want to have to deal with that. So in 4 days, it seems unlikely that a lot of those are going to be showing up. And so we do this, we keep them on ART for 6 months, and then we stop the ART and watch what happens. This has really sort of fallen out of favor because it was shown that this reservoir lasts in every animal for 6 months. A few animals out to a year, but if you go further out than that, there's nothing coming back. So it doesn't maybe represent the true reservoir. We've started to do studies now looking where we start ART at 6 or 8 weeks after there's immune responses, cytotoxic T cells, antibodies, and then we go out for a year. It's very frustrating because you set up this experiment and it's like, okay, get ready, guys. We get to look in a year. And every day is expensive in this world. So in the end, after a year and all of the scans and everything else, each animal is probably about $50,000 investment. So
How many animals do you make?
that's a good question. More. The answer is more. So this just illustrates the same sort of thing that I showed before. So we do these scans, we scan the tissues. We can put them in the blocks, and then in the hot blocks we can find the tissues, and the results are so sort of strange that I call it the rebound zone. And I hope this is going to work, but this was sometimes you got to make a point and you have to do something a little different to make the point. So here we go. Let's see what happens.
You unlock this door with the key of correlative PET scans. Beyond is another dimension, a dimension of anatomy, a dimension of physiology, a dimension of tissue. It is the middle ground between the reservoir and viremia of hearts and myeloid cells. You've just crossed over into the rebound zone.
If you're of the right age, that is a very accurate redo of that TV show from the 50s and 50s that they kept showing. I wasn't there at that point, but a couple of the shocking things is, one is we see these signals in the heart, and it's hard to sort of understand, but we think that it's being filtered, that we know that there are infected cells in the lymph, especially from the work of Mike Betts. We know no one can ever find infected cells in the blood, so they're getting filtered out. And I think one of the places they're being filtered out is the heart. And there's a lot of heart issues associated with HIV, even if somebody is on ART. The other, and we're working on that, the other part that was really making me insane is we can find these infected cells. In the early reservoir, we never see infected T cells. That's very anti-dogmatic. And then we could show that some of them were dendritic cells, some were macrophages. But a bunch of them we couldn't figure out what they were, and it was kind of driving me crazy. And so I'll go back to that in a second. But the thing that is sort of neat is that this rebound we think is reflecting the reservoir, and so we can follow this over time and quantify it. And so at 1 week, starting ART at day 4, we see these signals within an area of the gut. After 6 months on art, those signals are not completely gone, but almost completely gone. And then after you stop art, 4 days later, it goes off like a bomb, and the signal is greater than it was at any point previously. And so, and the most important thing is that this is saying, at least in some cases, there's an immobile reservoir, because we watch it fade away in a spot and it comes back in that spot. And if it's immobile, we know where we kind of know where it is, and we can dig down in there, and we're in the process of trying to figure out what that reservoir is right now. And so there was this conundrum of trying to understand what these cells were. They weren't dendritic cells, they weren't macrophages, and it turns out that they were in fact, mast cells. And I think this might be one of our most consequential rediscoveries, because if you look in the literature, there were 4 groups, including a group in Australia that published 9 papers. Group in Australia, including Tony Cunningham, published 2, and somehow that just faded away over time, like many things do in HIV. But we could show in the animals that there were sufficient markers, the FC receptor for IgE, the stem cell factor receptor, et cetera. These are infected mast cells. We were luckily able to identify collaborators at Northwestern that work with mast cells. These are primary mast cell cultures derived from human skin. If you kind of had a student or somebody in the lab and they said, my cells look weird, come and look at these, you would look at those. There are all these granules and stuff in them, and you'd be like, kill them and start again, because there's something really wrong. But this is what a mast cell looks like. And so you know, we have 2 models. There's a humanized mouse model, and you know, these cells are never in the blood, which explains part of the reason that they really haven't been missed by the HIV field, because the HIV field likes to focus on the blood, even though it's clearly the wrong place to look. Everyone says, okay, Tom, you know, do they make CD4? Yes, they do. Do they make CCR5? Yes, they do. And we can infect them in culture. These are different donor mast cells. I can talk later if anybody's interested about how you get these to go. And then there's some cell lines that are very close to being mast cells. They require stem cell factor, for instance, during culture, or they die like a real mast cell. And so we're kind of going through this and studying this, and I think this is going to be really quite important. But one of the neat things about finding these little pieces of tissue for the imaging is we can take sections from there and do other methods, the omics. And one of the things we're doing a lot lately are spatial transcriptomics. You know, when we started to do this, I wanted to do it because we were trying to figure out what the mast cells were, and we didn't know yet. So I was hoping this was going to provide some insights. It's actually, there's some limitations to this method, because you're just getting like little spots on your tissue, not the whole thing. So it's not single cell. But we began to look at early art, like what's happening at say week 3 and 7 of starting art. Early during the eclipse phase, when you stop it at 4 days, you don't see it at viremia, but yet it's we can detect it by the PET CT, and then sort of plastic rebound when the virus is returning in blood. And so this just kind of illustrates these pixels, and really what we're looking at here is, you know, depending on the spot, there can be 15 cells there can be 5, but it's a bunch of cells, and you do the RNA-seq on the cells in that area. We do pretty deep sequencing, but you know, to start to see things that are less than 1% or more than less than 1% becomes very hard. And so at least in this circumstance, and I didn't really appreciate this, but very much do now, is a 1% gene in 15 cells. You can see cell type specific genes more easily than if you do RNA-seq on the whole tissue, right? Because it's a low abundant cell and it's being dominated by others. You really don't see those signals. This just sort of illustrates we've now developed a bunch of methods. We can do multiplexing where we restain these tissues, and we have computer-aided identification of positive cells and overlapping signals that really make a difference. And so now, instead of looking at some small areas, we can look at an entire piece of tissue, getting thousands of cells and getting some really strong statistical data. And so along the way, you know, this is all new to me. I'm not a biologist, so I'm kind of picking up the lingo. So it's workflows and pipelines and atlas building. If it's the old Tom, it's stamp collecting and fishing expeditions. In the end, I'm tearing my hair out because we have so much data it's like you have a complete survey of 20,000 genes in each one of these spots. And it just is becoming a challenge, but at first glance, I think we're seeing some important things. And the key is, not necessarily the method, but the piece of tissue you look at. And so we have been able, because we have the piece of tissue where the virus is, we confirm that by PCR and staining. And then these red boxes here are areas where we found infected cells, and we can see, for instance, ISG15, an interferon-inducible gene that's known to be expressed in infected cells. We can see that that gene is turning on in most of these areas. We sort of gave up on trying to do everything in a single section. It just doesn't work, but we take subsequent sections. And so this is sort of an overlay. So it's not perfect, but it's pretty good. And so what we've been doing then, actually what Schwab and Ramon and Chris have been doing, and just really, it took a long time. These slides you do this on are like thousands of dollars each. And it's like, how'd it go? The piece of tissue floated away. So, you can use it again, right? And you're like, no. So, they really worked hard on this and refined this and have gotten really good at this. And so now we have 31 pieces of tissue that we're doing this with, and this just illustrates sort of the beginnings of some of this. This is a piece of gut tissue with an immune aggregate and this even more dense area. And so you define these clusters of gene expression that have patterns throughout the tissue. And so we can see patterns associated with these immune aggregates. We can look at the cells that are there and get a sense of some of the mucosal immunology that's going on. And then we can look at the gene expression patterns. And so here in these immune aggregates, what we can see is, what you would expect. Adaptive immune response, lymphocyte activation, et cetera, is in these sites. When you look at different areas, you see different sorts of signals. So here we're looking now in the yellow square in a place in the tissue where there are infected cells. And it has a different pattern. So not pattern there overlapping the infected cells. And what we can see is things like actin filament processing, cytoskeleton organization. It's like, what is that? You have to learn how to speak to systems biology. That's migrating cells. So there's migrating cells there. And then you have epithelial cell differentiation, these sort of viral process, other things. And that's sort of some of the repair and thing that takes place at these sites of infection, where the immune system is trying to come in and clean up and repair. So, and one of them that's very interesting is this viral life cycle that popped up, and it's things like TSG101, other genes that are turned on and activated cells that we know helps the virus to replicate. So I'll just kind of go through these quickly, but we have different versions of this, 31 of them all together. And so we can find the infected cells, we can characterize where they're at, we can see these different sorts of patterns. And again, we in these tissues, we're seeing after this, during our treatment, we're seeing these evidence of a tissue repair, et cetera, kind of at these sites. We can overlay where the infected cells are here. They never quite line up exactly right, but we have algorithms that can align them as best as possible. And then we can see what's happening underneath the infected cells, what's happening around the infected cells. And one of the when I was a student, we learned about interferon, and interferon is released from one cell to tell the cells around it, like there's a problem. But it seems like these days we're more obsessed with like the PAMPs and how is it recognizing it and what's the signal and focusing on what's happening in the infected cell. But this, looking at some of this, has really reminded me of the idea that you have this area, there's a cluster that we see of genes, differentially expressed genes that's associated with the infected cells. And then we see other patterns around them. And again, in this case now we're seeing these effects that are metabolic, but are related to RNA splicing, other things, again, in some of these tissues, some of these foci. And so looking proximal, what's interesting is the green cluster is not overlapping with the brown one here, which is where the infected cells are, but it's sort of around it. And we see some genes are overlapping, some genes are different, and to sort of put it all together here as I wind down, it's basically there are certain genes that are seen associated with infected cells or around them in all of the different circumstances, early rebound, late rebound during infection, and then there's other genes and patterns that are unique to each one of those. And we are doing our best to try to sort through all this. We see a very nice reproducibility within the same animals and these different clusters. And I think this is another example in science of basically the method getting in front of the analysis. So we and everyone else are struggling what to do with these mountains of data. But I think it keeps moving forward and the tools keep getting better, the computation keeps getting better. And so we're going to start to learn, I think, some very interesting and important things. So really, we can take these tissues and we can do imaging, the multi-scale imaging and things that I featured at the beginning. We can do transcriptomics on the whole piece of tissue. We can do the spatial transcriptomics. We pull the virus out that's there. We use barcoded viruses and we can get a sense of, is this one virus that is throughout this area? Is this multiple ones? Are there these accumulating dead genomes? Et cetera, et cetera. And altogether, I think we're going to really get some insights into what's going on with the idea of pushing this back further and further to get a sense of, right now we're looking at what happens during rebound and early rebound, but this is going to help set us up to find a reservoir. I got into trouble years ago. Meeting was getting a little boring and I raised my hand and I said, I think I know where the reservoir is. Ooh, where? I said, it's everywhere. And some people got really mad. But now everybody agrees it's everywhere, and it's different cell types, and it's a whole bunch of things, and it's going to be a challenge to finish this. So this is the group here, and I don't know where I'm at. See, I'm kind of long, but anyway, we kind of this is the front of my house during the pandemic. We took the Christmas lights and put them out there, and just very quickly, we developed culture systems that the upper respiratory tract is most similar to the upper female reproductive tract. It's columnar epithelium with cilia, and we could infect these first, sending them back and forth to Southern California to use their BSL-3, but they produce mucus. There's all of this activity there, and we can show that essentially. So here is we can culture these for 9 days, and you can see the first cells getting infected, and it doesn't spread, just diffusely. It's being dictated by the beating of the cilia and the structure. And it's kind of recapitulating what would happen in your upper respiratory tract to cough things up. And then I just love the picture. Ron Easy, Francois Billinger, myself, about 100 years of HIV, SIV research, and really the lesson is, during the pandemic. I have a big network. I know lots of people, and I had people giving me like COVID infected monkeys and stuff. And so we made a probe. We got some money and made an equivalent probe like to SIV for the coronavirus, and it works even better. We did a variety of studies. There was a whole bunch of limitations, and we were always last in line. But we were able to do some experiments, show this worked pretty well. I think this could be a great tool for long COVID. And one of the things that jumped out at us was that we could see infection in the male genital tract, the testes and the penis. This is unexpected, but in the end, there's a lot of ACE2 expressed in the testes, especially things like Viagra actually are working on the ACE2 axis there, and so it sort of made sense in the end. And what we could do is we could do multiple scans. So here we're looking at the lung. And so you have this diffuse signal, which is fluid in the lungs. I also like, learning how to read X-rays and all kinds of crazy stuff. And then you can see that the probe overlaps with that. Well, at 7 days later, the localized pneumonia has been resolved, and that signal is no longer there. At the same time, in the male genital tract, the signal is getting stronger. And so once we became convinced of this, we still are trying to get this published. Because the reviewers are like, oh, you need to do this 10 times. It's like, you don't know how much this costs, and there's no more money, and that ain't happening. So anyway, but we became confident enough and felt the world needed to know about this. And so we got picked up by the New York Times, and it was really crazy for a little bit. And we were all over the internet with all kinds of pictures. We were getting people calling us and contacting us like, who drew this picture? This is wrong. The testes are too big and the penis is too small. And it's like, this is a monkey, and we introduced some YouTube videos to illustrate things. and I get this thing from YouTube that's like you're banned and whatever, and it's like, for what? So I you know, but you could appeal. So I write this very long appeal stuff. The algorithm comes back like 2 seconds later, no, appeal denied. But it turned out that it wasn't our video, it was the 600 things in the chat underneath of people arguing about vaccines, which I never got to see because they deleted it all. So anyway, you know, I think my best story from the pandemic was I got canceled on YouTube, and I'm putting that on my CV. So, and then there was a lot of outreach, and it was interesting, and I thought this little clip from a Sunday paper in Miami said it all, right? It's like, you know, while most vociferous anti-vaxxers declare their willingness to die rather than accept their jab, how many are willing to live without their junk? So that sort of changed everything, and you know, I actually know quite a few people that got vaccinated. And so that was my little contribution, my last contribution. I am very proud of that. So thanks for allowing me to kind of throw in that little bit at the end. And sorry, I went a bit long. I'd be glad to try to answer any questions. Thank you so much for your time. Oh, sure.
That was just amazing. And engineering antibodies, where they are.
And it's just getting interesting now.
And the Omics. So, any question? I'm sure there are many. Go ahead, Heidi.
Thanks, Tom. That was terrific. I wanted, in the first part of your talk, whether you looked at different isotypes of the same antibody.
So we are in the process of doing that now. We are, alas, we're not antibody makers. It's a challenge, especially for the amounts here. We're sort of antibody panhandlers, but we have gotten some resources, and there are some companies that, for instance, for BRCO1, I know that the person doing this is, I saw she was watching this, but Ann Crias, but we have IgG1, 2, 3, and 4. We want to start to look at some other things like IgAs, and we just have to begin to sort out those details. So we're just getting going on that, but I think there's lots of interesting possibilities.
Yeah,
So
I guess I'm just interested in the DIGA traffic to the places you actually want it to protect against HIV more effectively than IgG.
right. So the biggest challenge in the dimeric IgA, and I think we're missing some really important possibilities, is when you make it in a tissue culture dish, it's just this very heterogeneous mess. So so we need to come up with better ways to make the dimeric IgA and to watch it. But there's been some heroic efforts, one of them by Sally Permar, where she IV injected dimeric IgA and showed that it ended up in breast milk, for instance. So we are very eager to do those kinds of experiments and just sort of getting going. And we have recently gotten the resources we need to begin to look at this in some detail. So there's a bunch of neat stuff to study.
Meetings online. The chat, but I can't see that question, but I'm guessing.
Correct.
Mast cells, maybe asking this.
Mast cells are concentrated at all places where our tissue is exposed to the outside world. So for instance, they're really present just under your skin, they're present in the lungs, and they're really concentrated in the gut. And they're also like very rare. There's hardly any of them around. So we have found, I think, a few infected mast cells early on. So, but they're not major players in transmission. But I think they're going to. We find them very overrepresented at sites of rebound, and they are known to sort of accumulate at places where there's an insult. Whether it be a physical. Something, a bruise or something, the mast cells will come in there, an infection, they'll come in there. The best work is looking at inflammatory bowel disease and syndrome. And usually when you have the flare-ups, after you get past the sort of crisis, it's in the same place and it's sort of correlated with these mast cells that are there. So, mast cells are fascinating, and people have been fighting with mast cells forever. We all take Benadryl, Zyrtec, all of these things work on mast cells. There's a bunch of therapies that are developed for mast cells, tyrosine kinases, antibodies that cause their ablation, et cetera, et cetera. It's kind of unexplored territory in HIV. And we have this neat potential to take things that people use every day that are in the clinic and then ask these other sort of questions. So we have a nice program project grant to try to study this. We're very interested in looking at and trying to understand it. But they're kind of like, if you look in a tissue section, you might see like a few of them because they're just really spread out and few and far between, and they are not in the blood. If you have a mast cell in your blood, you have leukemia.
So how are you sure that they don't have anything to do with transmission? Because are they producing virus?
So they do produce virus. You know, I showed early on, and I don't really want to go back, but there's just very few of them. So I can't say that they're not getting infected early on, but what we can say is 80% of the infected cells in a with even a replication defective vector are Th17s. And then you know, 75 to 80% are that. Then, you know, then most of the rest of that is immature dendritic cells. And then there's this tiny little bit that are others. We've been looking at this, and I'm sure we'll find a couple of them, but that's very different than when we look at one of these rebound sites, and in a one piece of tissue, we can find 40, 50 infected mast cells right all there together. And again, it's striking because you should not see 40 or 50 mast cells in your average piece of tissue. They're overrepresented there. So now, are they the tinder that allows it to go off like a bomb? And what's neat is when we look at the later reservoir, you know, starting art at 6 or 8 weeks, where we have cytotoxic T cells, we have antibodies, the bomb doesn't go off. It was like, what? So that's a great question. And in mice, they can live for, they divide in the presence of stem cell factor. They expand and contract depending on the local environment. So a clone essentially can persist for the lifetime of a mouse. Whether that's true in a human isn't clear, but you know, half of what we think we know is wrong, and straight up wrong, and then the other half we have no idea about. So you should question dogma all the time, and a whole bunch of these things were sorted out decades ago, and everyone's like, oh yeah, blah blah blah. It's like, no. And, go back and look at it again.
No, like another question that I was thinking about. Have you tried antibodies that like from camels or bovines that have like they say that they are like super neutralizing antibodies, and they have like this long
Right.
CBI.
So I mean, I think the antibody binding to the antigens is not really a limiting thing at this point. You need we're binding to you know, we might bind to viral particles, but mostly what we're seeing is infected cells, and the envelope, the SIV HIV envelope is in a different configuration. So if some antibodies bind that well, especially the CD4 binding site ones, bind really quite well there. We are about to start to look at some of these monobodies from the llamas and camels. We're actually doing the first experiment coming up this week. And they're also smaller, so maybe they can get distributed better. So one of our biggest things we want to do is get the probe into the brain, and that is everybody wants to get antibodies into the brain, and so that's why we're sort of going to start pushing on that. And maybe a smaller protein can get in there better. So we're very interested in looking at that, and we're always trying to improve our methods. But with these FAB2s, we're able to find, my team has just gotten really good at every single animal. We can find tissues and go in and find virus and infected cells and all that. So we got to improve it, but that is sort of in process.
I'll make this last question because we do have tea in the back.
There's also one.
Yeah, well, I can keep talking. It's okay. We can get to you and keep talking.
Come in the chat.
Well, no, go ahead. He's been waiting.
Oh yeah, so just, I don't know, a little big picture question about like the reservoir and the cure. So presumably the contact lesion, the idea is that when you find the cells, you can target them, then you can kill your reservoirs, right? But it's saying that the reservoir is everywhere, it's all over the body. So I'm wondering about the magnitude of the size of this reservoir and what impact that might have on, you know, if you're trying to kill cells that are distributed all over the body, if there's enough of them, that might maybe be a problem for the person that you're trying to cure. I wonder if you have any comments about that.
Well, no, so what's interesting is, you know, that basically one virus or one lineage tends to grow out when you stop ART. And so that was believed to be compared to transmission. So it's like it starts with one virus. But in fact, that one that emerges now, it's very clear it is like dodging cytotoxic T cells, it's dodging antibodies, and it's dodging innate responses. So we don't have to stop them all. The body's got kind of in front, your defense systems that evolution gave us can kind of get control a bunch of it. It's the ones that squeeze through. There's some neat human studies where the virus that comes back when you stop ART is very often the virus that was there when you started ART, and that's because that virus that is dominating is in the fight with the immune system's responding, and then when you start ART, you sort of like freeze that in time, and then when you stop it, it kind of continues. So we don't have to get rid of everything to have success, and there are ways that the body is controlling some of these things, and this is I say lots of crazy things. Anyway, I think the mast cells are the reservoir that we're looking for, and everyone's like, oh, look, a mast cell, can't be that. It's that T cell in the corner over there, and that's starting to stink a little bit, I think. The whole T cell, latent T cell, it's not a latent T cell anymore, it's a mostly latent, sometimes expressing, and really, do we need to even do these things? There's pathology, even on ART with totally suppressed virus, there's these biomarkers, IL-6, et cetera, that correlate with disease and cardiovascular problems, all these other things. Maybe what we got to do is get rid of the source of the IL-6. It's not T cells, they don't make the IL-6. It could be macrophages, but who makes a lot of IL-6? Anybody want to guess? Mast cells, that's right. So, breakthroughs in science don't come from somebody who did the same thing for 30 years and continues to block other people from doing other things, et cetera. It comes from the young people here, new ideas, new approaches. The future belongs to you. And listen to what everybody says and then do something else. Because as a young person, you can't compete with those established labs anyway. They're going to block you. And then you do something very different. I think you have a much better chance of success, and they will ignore you until it's too late. And then they'll try to catch up, and you just sort of, you should have figured this out a while ago. So, so there it is. Sorry. Okay. So, we do detect, I'm sorry, but the virus we're detecting early on those lymph nodes is probably associated with dendritic cells and other cells that are ferrying it there. And so we have to look at that more clearly. So I'm not sure if this is a method of virus transmission, but it's just a method of how antigens are collected and processed in the mucosal environment of the gut. And if you look in textbooks and things, that is kind of how they will describe it. It's these CD123 dendritic cells, and we can in fact show them full of virus at these sites. I need to kind of move that into it. And yes, so another mast cell enthusiast. I'm trying to just get people excited about mast cells because this is a job that is way beyond one lab or one person. So if we can get 10 labs or 20 labs kind of looking at this, I think that we have a chance. And again, this was discovered before. We sort of found it again, and just think about it. And the mast cell world is insane, actually, as you start to go down the various rabbit holes. People think it's the basis of autism. People think it's the basis of all kinds of things. But mast cells are, they play a huge role in the gut-brain axis. They sit at the ends of these nerves, and they pick up these neurotransmitters, and they emit them, and they're playing all these kinds of different roles. Mast cells are the remnants of the immune system before VDJ recombination. And so as adaptive immunity sort of emerged, they were there, and then the partners of these mast cells are the cells that drive me crazy. They're all these low abundance cells like ILCs, ILC1s, ILC2s. These are echoes of the old immune system, and as the new one came along, they provided some functions that, didn't need to be replaced or they didn't have to be replaced. And so a lot of these low abundance cells are there. The whole Th1, Th2, and again, there's connections with HIV. The Th2 stuff is mast cells and these ILCs and all this Tregs and all these other things. So go read about mast cells and get ready, because you're going to get drawn into some interesting things, and it'll just make your head spin. But I really feel there's some missing piece in HIV, and maybe there's a chance that this is the missing piece. So anyway, I see that Lindy is talking with Ann here on the chat, so that's kind of good. And anyway, sorry, another question? So yeah, that's great. No, it's time to wrap up. Thank you so much, everyone.